Come on. Come on. Give me some.
It's like...
Fantastic. So I quickly wanted to tell you about something that me and my colleagues have been working on that I hope that maybe some of you will contribute to or maybe benefit from. I wonder, is it just my laptop that's weird?
You have to be careful with that cable. It's super...
Well, it's like every cable.
Yeah, would somebody come here and just hold this down like that?
Put something on there.
Put the remote on it. Oh, there it is.
Yeah, it's not heavy enough. Just do like that. It won't be long. All right. So I have a tendency to create ultra long URLs. So I'm often surprised at the degree to which psychological experiments like the stimuli scripts are not shared. There are great, fantastic open source tools like PsychoPy, VisionEgg, and even some MATLAB tools that allow you to reuse things that are produced in other labs. But if you can imagine, I would probably estimate that the MBAC task has been re-implemented 8,000 times, if not 8 million times. So what I wanted to do is create a repository for storing these. I couldn't find a very good repository that wasn't tool specific. So for example, there are some repositories for presentation or from other things that are specific. So the goal is to be a generic one that has tools from a lot of different things. If you see really quickly, so at this point I'm the only one that's contributed to them. But for the tools, you know, our plan or our hope is people will have it in a GitHub repository, submit it to Zenodo for a DOI and track it through there, and then here we have the basic sort of reference information for them to have, and then we ask that individuals link to their GitHub repository, and then in the README have all the information to help with the task. But anyway, it's a resource we're hoping to build, so if y'all have scripts lying around, please contribute. Thank you. Oh, sorry. Open Cog Lab repository.
I was sure on the lightning talk thing.
.github.io.
I thought there was something similar from Rust bugs. You're trying to compile, load it.
Yeah, so this, I think what we're doing is a little bit different. So do you know about Experiment Factory? Have you seen that?
Oh, right.
So that is to create a universal way of specifying the experiments. Is that right?
I mean, I think it synergizes with this, right? So what Experiment Factory does is it basically lets you just drop in new experiments that have been written in JS Psych, and then they'll just show up, you know, in the database, and you can run them online. So we could imagine taking, presumably as long as we had, you know, a GitHub page with a DOI for each of those, they could plug into here and they would just show up as an experiment.
That would be fantastic, yeah. And we also created a Zenodo category channel. So if y'all are submitting to Zenodo, y'all can put it in there.
So this for like IO, it is. I don't know why that didn't work. It's OpenKeg. It's not going to work. That can have .io.
Does anybody have it? Oh. This thing is amazing.
That would be just.
Yep. Oh, you got it.
All right, hi everybody. My name is Matteo, and I think that most of you have already seen this presentation given by Jarek. But if some of you haven't, I just want to show you this tool that we developed actually at a brain hack 2 years ago at OHPM. And the development has been going on for a while, and I just want to talk to you about with that. But first I want to start with like some rhetorical questions, like who writes their own methods? Who uses methods of others here? Who would like their own methods being cited? Who cites the methods of other people? All of them? Exactly. So this is the problem that we're trying to address. That is, methods, softwares, and data sets are often not cited adequately. And if they're cited, actually the version is often omitted, and so it becomes a problem for reproducibility and everything else. And to be fair, it's not that we're just evil. The problem is that it's really tedious to collect all the information needed to do to reference these. And I think one of the problems is also that the publication system doesn't really help with this, because oftentimes you have a limit of the number of publications that you can cite, and then unfortunately the methods are the first ones that you cut, because you think that like who's going to read the paper is not interested. But anyway, our approach, and if you haven't met Jarek, this is Jarek, but I think you all met Jarek. Our approach is to make it very easy to collect the references for method, software, and the data that you used in analysis pipeline. And the idea is that you have your own project, and while you're running your analysis, then Craddock will actually accumulate the information of all the methods that you used, so that at the end you can just type a command and get all the references of whatever methods you have used. And we also try to make it easy so that you can output the references in the format that you like. Ideally, you can just use a style format and get it in any like Chicago style, APA, etc., or LaTeX and BibTeX. So actually I was very surprised when I did this very small example, because as you see, so this is just basically 4 lines of code, 6 lines of code. It's just making up some blobs, clustering them, and printing done. And you can imagine it's very easy. Like how many citations would you need for this? And so when you run Craddock on this tiny example, actually tells you that you should cite 3 papers already. And actually you can just run it by importing the module, Craddock, and running it here, and it will say, it will give you this report and tell you, blah, blah, we use NumPy, SciPy, and into SciPy, we actually use this method, and then you can see all the original references. But this was a very tiny example. If you actually use a bigger example, which comes from a from the test suites in PyMVPA, you actually see that we should actually be citing a lot of methods that probably we're not citing that often. And what if we want it in BibTeX? Well, you can just type the credit summary, format BibTeX, and you get all the references that you can just copy and paste in your bib and then use it. So how do you do it? Well, it's actually very easy. Actually, for the project that we used, I already that we are working on, I already put this tab so that we can just, as we develop, we can put all these references. And the idea is that you can just copy this tab into your main project, and then in every in every module or file that you want to use, you just import these 2 things. And the nice thing is that you can refer to your, actually to the papers either with a DOI, and it will automatically fetch the BibTeX, or you can just put BibTeX, and we're working on extending the way that you can reference to these papers. And this is in your software. Alternatively, you can inject these references into existing packages by creating, for example, this comes from a SciPy injection, so we're not touching anything into SciPy, we're just putting these into our Craddock project. And the idea is that by doing this, we're not modifying existing packages, but we're just basically achieving the same goal without having to touch it. And please get involved. So if you want to start using it, I know that Ariel used it in Shablona. Please report bugs, because as you can imagine, we don't want to be a problem for the existing packages, and so we want to make sure that everything is tested and works correctly. We have some ideas to extend this to other languages like MATLAB and Octave, and somebody is working on that. But then it would be cool to also implement this in Java, R, C, and C++, but I have no idea how to do that. So if you have ideas, that would be awesome. And spread the word. Thank you.
spark corridors being just merged into NiFi, and they're slowly but surely propagating NiFi interfaces, printing them with references. So then you can run it through uCredit module and will give you all the papers that you decide.
Thanks. Yeah.
What's your Twitter handle?
At MVDOC. I have a long last name.
All right.
Any tips for how to do this? You have to restart at this point.
Do you have something like a shortcut with which you can usually function and some window? Just one of those, maybe this one?
It doesn't detect it even.
Can you try
Nice thing is
Fn and F11?
that are all sort of deliverables in their own right.
No.
No. Which thing you used? Which cable?
Here, the HDMI.
Building an open source version of the algorithm that was inspired by.
Are you connected to the wireless or wired?
The wireless
Through
doesn't work.
wire.
It's sort
Oh,
of
the wireless.
just like a graph.
Do you have
right? Because there's all kinds of problems in different. Like,
do you have other outlets? Other outlets?
Mission using all sorts of modalities. So like that's one.
Or you just have HDMI?
It's just HDMI. I'll restart because it might just be enough to restart.
Because
And then ultimately.
We have just a behavioral.
yeah, I don't know. I mean, sometimes you just need to redetect, but since it wasn't even funny. You're on Ubuntu.
But the cool thing is that we can build a framework and just over time, like more and more
That's fine.
variable gets annotated, or we can go.
I mean, most of it's more of an index than it is
The stuff's
specific.
there. So it's,
So,
yeah,
I mean, we prefer it.
think that in terms of how
Tools if you use that.
Oh, we can change it.
horizontal we can integrate it.
We can try the searching.
Right, as if I thought of it as a much more vertical project. Right,
After
whereas
you.
like we do this and we do
Yeah,
this.
if you need help,
this,
it's
it's extra
shit.
expense.
Yeah, it's essentially.
Let's try it.
It should be an HDMI.
Working on it on GitHub, so it was like, oh, like this library that you're writing, like, is like, should be a sub-module in our thing, but it doesn't actually need to be, we can pull the like G-Fusion out of Confusion, right? Because if, you know, you run engineering problems in Graph Fusion, you don't use it by definition. You want to clone like all the graph we do.
Alright, so at this point maybe it's better to borrow someone's laptop and then just excellent.
You only have 63 minutes left. No. Jasper is just bolted on like engineering web service. It's like I've actually learned how that works. Maybe Jasper's was working.
No, it's a Mac.
And now,
What's the other one?
I think when we cycled it, they got out of sync.
Excellent, thanks, Matteo. All right, so about NiProf, here are some instructions if you want to execute these commands. I won't be able to, follow you through. But it's basically just about installing NiProf. There's some sample data there that you can run, and if you want to have a more detailed look at images, you need these dependencies down here. So you've all heard about the importance of reproducibility, and we've heard people talk about how pipelines improve reproducibility and through a software way. But another way to look at it is look at the metadata of files, the actual brain images that we store. In our lab, we ran into a situation where 4 to 5 research scientists and postdocs were all saving files on the same computer. So we had like 10 to 20,000 brain images on the same computer, and we were kind of getting lost. And now and then you would be trying to make a figure for a publication, and you would not be sure anymore which filter was applied at what point in the analysis, or which subset of subjects was in this image, etc., etc. So we were thinking about how to do this, and we decided to just start keeping track of the brain images that we create, and how they were created, and from what parent images they were created. So that's what NiProf was made to do. So it's a Python tool. And we think that it's too much work to track this stuff by hand. So we need an easy way to collect this metadata alongside your regular pipeline steps. So here's after you've installed it, you can run these commands to collect metadata on your images. So if we have a directory called test data, which is in the package that you can download, it will show you when it's importing these data. You should imagine that you're running this on your main data directory. And then you can look at the metadata for these files using the provenance export command. It just shows you a list, or you can look at one particular file, and it will show you things like the echo time or the repetition time for these files. This is only supposed to be done on the raw data, because we haven't done any transformations on those files. And then in the next step, you would run your regular transformation commands. For example, here there's a parec2nifty command or an FSL registration command. And you basically wrap those in a provenance record command. And the provenance record command will try to detect what images are created and what commands they are created with, and gathers a ton of metadata from those commands and from the images themselves. So after you've created a new transformed image, in this example is t1_reg.nifty, that will have metadata associated with it that was only obtained from the raw image. So Nifty images don't store things like the echo time or repetition time or what subject was used or what project this file is associated with, etc., etc. So we now have that in the database. If you use the provenance serve command, it will start up a little web server on your computer, and in a web browser you can start browsing through this data. I won't be able to show it to you now, but imagine, an interface. Yeah.
What's the parent new? And that the example with the parec?
Parec2nifty is a converter that's in a nibabble package.
The -- parent and -- new. Is this the parameter
right.
to provenance, or is this parameter to parec?
That's to provenance, yeah. So the second one, the McPhert one, it's able to tell what goes in and what goes out, because in and out make a lot of sense. But the parec2nifty file is kind of ambiguous, so we have to specify them. So it's a little bit more work in this case.
Okay, so the command lines are parsed, and
yes.
there are inputs and what are outputs differ from the command lines.
Yeah, exactly.
So if I do some, God forbid, free filter that take subject's year and subject's name, then I'll have to annotate them with individual files that they...
So a subject name at that point provenance should know, because it knows from what raw image you got it, and the raw images have metadata associated that has the subject name. So you won't have to tell it about the subject name.
Is there a way that you can like sort of mask the environment? Right? So the idea that you would like activate a provenance session rather than having to like wrap every command in a provenance.
Yeah, you could make an alias if it's a command line thing. And so the next part is about Python. And in Python, that's exactly what I'm doing in this line here. The provenance context is just a provenance object, and it holds like a connection to the database. It caches some stuff, things like that.
Okay.
And then, yeah.
What about using strays or ptrace to actually figure out which files commands access?
Ptrace? Oh, trace.
Yeah.
That's what I'm doing here. So in the Python API, when you give the provenance record command a piece of Python code, like a function, it will see where that file is, the code file. It will see what the git commit is of that file. It will see, what our dependencies are there, etc.
And this is done through trace?
Through Python package utilities, etc. Not the system trace. Yeah.
Because there's this other thing called reprozip. I don't know if you heard about it.
Reprozip?
Reprozip. Reproducibility zip.
Okay.
So the approach is whatever command you run, it uses low-level kernel utilities for tracing all system calls, including which files are being opened, which files are being created, which files are being written to. And then it can capture all of that into a container and make it fully reproducible. But a similar strategy could help you to figure out, instead of parsing the command lines, to figure out which files have been accessed, which files have been created. And this way you can figure out inputs and outputs. There is a performance overhead though.
Yeah, I see what you mean. So here are some things that you can do with the metadata once you have it. There's a little utility that shows you snapshots. So you can browse through, for example, all the files for a certain subject, for a certain modality. We have one researcher who really likes to look at all files for a certain project, and then subjects with a certain age for that project, for example. And then this just loops through that and shows you snapshots of those images. And the other functionality here, the compare functionality, will show you the metadata for two images side by side, and will tell you if there are things that have been done differently between those two files. So if you're not sure what did I do on version 2 versus version 1, it will tell you what the differences are. I think I should leave it there.
Okay.
Other questions?
Great.
Where's the microphone? Is that over there? Oh yeah, it's down. On the bottom. It's probably okay. So we have a chance of this. I want to put it up.
Sure.
Somewhere around here.
Where are the wires? Alright, let's do all the fun. That's what there should be. Okay. Yeah, whatever. I don't need that. It'd be nice if I could use both and not break it. I was trying to make them both work, but apparently made neither of them work. That's cool.
HD, it's not HD.
Oh, it's what, sorry?
HD base.
HD base. There's at least one.
The one workspace.
Sure. Okay. So I'll tell you about some work that I've done recently, and the code to do the calculations are on GitHub, and I'll give you a link to that at the end after you know what it's good for. So I've been using a measure called fractal dimensionality to look at brain morphology. So I've been doing this work with Elizabeth Kensinger at Boston College. We have funding from places, and pretty much all the data I've been working with is open access data that we've been talking about this whole time anyway. So what is fractal dimensionality? The idea is using a kind of mathematical approach to get a single number of how complex a structure is. So the yellow thing here is a fractal. How can we measure how complex this thing is? The idea is to, let's say, count, let's say, all of the red squares and see how many there are relative to the size of kind of the graph paper we have. We could change the resolution of that graph paper, we get a different number. That would just be a kind of the edge or of the surface of a, say, 3D structure. The code and the analyses I've done have looked at both the surface and the filled volume. Filled volume stuff works better, so I'll kind of just focus on that right now. So in that case, I'd be counting all of the red squares and all the yellow squares and seeing how many there are relative to the size of effectively the graph paper. What does this look like with, let's say, a brain or, an example first. This is Germany. We can look and see across different kind of grid scales and resolution how many of these squares we'd have at a given resolution. If you plotted that in space, you'd get that kind of exponential function on the side. The dots will always fall exactly along a function. If you go in log space, you get a straight line. The slope of that is the fractal dimensionality of, here, a 2D structure. If we, I made these slides earlier today. Why do we care? Well, prior to the work that I had done, it had been shown that with healthy versus people's brains that of individuals with pathological or psychiatric conditions, this kind of measure differed. So, borrowing from this approach of just how do we compute the quantity of how complex a natural structure is, we borrowed that from fractal dimensionality. Brains. So on the leftmost, just below the 1, that's the original 1 millimeter isotropic volume of brain. We just are looking at the gray matter here, so just the cortex, like the cortical ribbon. This is the highest resolution kind of on the left. If we kind of use different size graph paper and just look at how many squares are there at that size, it gets more blocky, and there are less of them, but it's a way to see how does the brain change across this. There is the issue of using this kind of graph paper approach, which is the original way. How the graph paper aligns with the structure will give you slightly different answers. Like if you just shift it over less than a graph paper unit, that's not great. So instead I use 3D convolutions to kind of smooth the structure and effectively dilate it, and then you don't have that problem. So for this image, which is from an actual subject's brain, we can look at it, we get a fractal dimensionality value for them. This person happens to be a 35-year-old female. But what if we look and give it more than one brain and look across and see, for instance, with aging and across lifespan, how does this differ? So I'll try and not use the word change because the data is cross-sectional, so how are age-related differences in fractal dimensionality? So if we just look at examples from a large data set, these are from the ICSI data set, IXI. Some images at the top, these are brains that had high complexity scores, lower is brains that had low complexity scores. They look different, and also they end up happening to be people, people in their 20s, people in their 80s. So it does seem to have some correspondence with age just at a course level here, and they do look different. So whatever these numbers are coming up with is somewhat visually identifiable. Here are spinning brains of 2 of those subjects. As you can see, the one on the right, who's like a 70s, the gyri have more, the gyri themselves aren't as wide, the cells I have broadened, versus the other brain, which is more kind of tightly packed cortex. So what changes do we know that happen with in brain morphology? We know that there's cortical thinning. We know there's some changes in gyrification, subcortical volume shrink, ventricles enlarge. Most of these measures are kind of more volumetric apart from gyrification, but what about the actual shape of structures, which is something that this complexity measure is capturing versus just like the distance between two surfaces or like literally like cubic volume. So I use a standard free surfer pipeline and then do parcellations. For now, I'll just say cortical. I did do some subcortical stuff as well. And just effectively using stuff from the cortex. So if I just took whole brain mean cortical thickness, we get this plot here. Fractal dimensionality is our y-axis. It looks like this thing relates. If we look across the lobes to see if there's any differences across it, or if it's kind of homogeneous across the whole brain, there do seem to be some differences. And if we kind of just take all this together and also looking at cortical thickness and gyrification from these same individuals, if we just, the ribbon being mean cortical thickness across the whole brain, we do find a relationship between changes in cortical thickness, well, differences in cortical thickness and age, less so for gyrification, but still enough. Complexity seems to do better than that. If we look across the lobes, for thickness we see mainly in the frontal and temporals where those changes occur. For gyrification, it's most pronounced in parietal, which does replicate prior research. Complexity gets more than they do. So that stuff I'll publish, and I'll have the reference come up again. Stuff that I've been doing more recently is, let's say, seeing if I'm going to say that this is a great mesh and people should use it, how reliable is it? So some data I've used from CORE. So the CCBD data set is one of the data sets within the CORE large aggregate data sets. GSP is the Genome Superstruct project, which has been run in the Boston area and is headed by Randy Buckner. Cortical thickness, it's reliable. It compares, these are similar to prior work. Fractal dimensionality seems to do better than that though, which is kind of cool. If we look at subcortical volumes, we can look at volume, IZV corrected, how that relates with age. Pattern generally makes sense. Complexity generally does better or the same. Interestingly though, if we look at the ventricles, we don't do better. So maybe there's this is some kind of evidence suggesting that actually what is the structure we're looking at, the shape of that matters, rather than just all structures are being, age-related differences in structures being measured better by this, but actually if it's, say, neural tissue versus CSF. So this does seem to be more sensitive to age-related differences than previous measures that have been used more often. I'm not the first one to use fractal dimensionality, but I'm using it here with age, which hasn't been done before. And possibly more usefully, I made a toolbox for it that basically just uses intermediate files that FreeSurfer produces anyway and gives you a tabular limited text file similar to what FreeSurfer would give you. It's on GitHub. Feel free to use it. If you do, please cite. And yeah, any questions? Yeah.
Would you mind? Would you contribute for CORE and the others, the FreeSurfer reconstructions you have?
Sure.
The pre-processed connectome project?
Sure. Yeah.
Load it up into the cloud?
Yeah. I ran all of them and then I figured, well, I probably just have been downloading all the open data I can, FreeSurfer it, and then figure out later, is this useful for things that I like, more research directed, but all the things are there. So,
well, the benefit is you'll have free long-term storage for it.
Right. So the main thing that I haven't done yet with, let's say, all of CORE versus the subset I've used is actually skipping through the surface and like doing QA, which maybe mind control will end up being useful for as an intermediate to do QA.
Other people too, maybe.
Right. Yeah, but like they've all been run, and extra measures in the case of fractal dimensionality beyond and gyrification beyond, let's say, standard FreeSurfer pipeline as well.
Fantastic. That'd be awesome.
Yeah.
Let's talk.
So if you put thickness and gyrification and fractals into the same model, how much unique variance do the fractals have?
Significantly more, and it captures all of those too. I don't remember exactly how much more. It actually is in a table in that paper. Like I ran linear and quadratic effects of all of these three, both whole brain and lobe-wise. It is significantly more that it contributes, and it captures all of thickness and gyrification. But how much more, I don't remember. Yeah. Anything else? Okay. Cool.
to mic up, right? So I'm going to do a quick 2-for-1. I'll show you one thing really quickly, and then I'll talk about something else without taking more than my allotted time, hopefully. Okay, so this is a package called Bambi, which you can find at github.com/Bambinos, and I know that Bambini is the plural of Bambino, but it was taken, so it's the backup. So what this is a package for mixed effects modeling within a Bayesian framework. So there are lots of Bayesian packages for Python, but none of them let you do things like this, where you can specify LME4-like models. So the idea here is this sits on top of something called PyMC3, which does all the hard work, and it lets you specify the very similar syntax to what you've seen in R, probably, models that include random effects, and then you can generate plots. You can also specify priors in all kinds of different ways, and via PyMC3 you can generate plots that look like this, so you get nice Bayesian estimates instead of just the point estimates and standard errors that you do normally. So you can take a look at that and read the documentation. But the thing I want to talk about is this, which is a paper I've been working on with my postdoc Jake and Tom Nichols, and it's about something called the stimulus as fixed effects fallacy. How many people have heard that term in psychology in some context? Okay. How many people do fMRI? Task-based fMRI? Okay, most people. Okay, so this is what you would call, I think, the standard model, which is also incorrect, it turns out. Well, usually incorrect. So the idea here is you have some participants and some stimuli that are nested in conditions. So the colors code different conditions. And this is participant 1, 2, 3. And the idea is that most of the time we have, or much of the time we have different stimuli within our conditions. But the way we tend to model them, in fact, almost universally in fMRI data analysis, is as if the stimuli have constant amplitudes, right? The stimuli don't vary. We pretend essentially that once we've lumped, these 10 stimuli into one condition, 10 to the other, there's no variation there. And this actually turns out to be a big problem because, in fact, there is variance associated with stimulus. And if you don't account for that properly, then you're basically not adjusting the uncertainty properly. So it might look like there's a big difference between this condition and this condition, but actually it's because there's one stimulus that happened to be particularly strong in this condition, right? So it's a stimulus effect that you're attributing to a condition difference, which is what you're interested in. So the fixed effect ends up the statistic ends up being inflated, potentially considerably. And this is an issue that's been recognized in many domains of psychology. This is what you'd like to do. You'd like to have models that allow stimuli to vary randomly, and you model stimuli as if they're sampled randomly. So there's a variance component for the uncertainty associated with sampling randomly from stimuli, just like we model subject, right? So everybody models subject as a random variable. We assume that subjects are randomly sampled from some greater population. Most people, I think, virtually everyone, in fact, acts as if we're doing the same thing for stimuli. We don't care just about the 3 stimuli that we sample. We're trying to make arguments about, this class of images, that class of images. So in this paper, we fit these models and kind of ask what happens to typical statistics when you do this. And here's one example. This is real data just to show you. This is the HCP emotion task. You take the amygdala, which in the ordinary analysis, you can see it responds to some degree to shapes, and much more to fearful faces and angry faces. And in fact, if you just looked at this, you would think, oh, look, it looks like the amygdala is more responsive to angry faces than to fearful faces. Problem is there are only, I think, something like 10 stimuli in each of these classes, and that uncertainty is not being accounted for. When you include the random effects in your model, point estimates don't change, but the uncertainty around them do, and all of a sudden, there's still a difference between shapes and faces, but no effect of valence, right? And this is if you want to argue, if you want to make claims about classes of stimuli, which I think we do, and not the particular images that were presented, that's the correct model. And here you have no basis for saying that the amygdala distinguishes between fearful faces and angry faces. So that's one particular example, just to give you a sense of what's going on. This is how you would normally think of the data, right? So you have stimuli in columns, but you can't see them. We don't model them. The rows are subjects. But actually, you can see that if you actually do model the variance associated with stimuli, there's huge amounts of variation. In fact, in many cases, the variance associated with stimuli is bigger than the variance associated with the subject. And that gets aliased often into the test statistic in the fixed effects. This is just showing you the posterior estimates. So these are individual faces, right? So there's variability in these effects. And so here's really the sort of, I think, the important point. When we did this for several different data sets, different HCP tasks, a couple of other data sets, on average, test statistics are reduced by half to 2/3. It varies across data sets and depends on other things I won't talk about. But you can see here that on the x-axis of each of these scatter plots across 100 regions, these are the sort of correctly modeled test statistics. These are the original ones. Did I break something? Okay, and you can see what happens, right? So this is what you normally would see reported. Actually, you should be reporting these statistics. There's a big difference. So let me just quickly conclude with probably the two questions and answers to them that are asked most often. How big a problem is this? So Jake went through 100 randomly selected studies, and the estimate is that at least 60% of fMRI studies. This is actually a lower bound because many studies couldn't, so these are the proportion of studies that could have and should have fit a model like this. Many studies can't fit a model like this only because they didn't have more than one stimulus, right? So you can't model variability if there's only one stimulus. That doesn't mean that you can take that as, you can generalize from it, then you shouldn't say anything at all about the class of things, right? If you only have one stimulus, by definition, you can't generalize. So this is a lower bound. I would say the vast majority of studies that use multiple stimuli or should use multiple stimuli. What do you do about this? Ideally, you include random stimulus effects like you saw. The problem is that there are no packages that are, FSL, SPM, et cetera, don't do this right now. And it's not clear what it would take to make them do it because it's a difficult model to fit. As you saw, we did it on a region basis because doing this voxel-wise would be kind of a nightmare. But at some point in the near future, hopefully, some package will implement the ability to do this in an efficient way. In the meantime, what's almost as good, it's not ideal, but it's sort of an approximate solution, is to use lots of stimuli, right? So don't use 4 stimuli in one condition, 4 stimuli in another. Use as many stimuli as you can cram in there, and you can use partially cross designs. That's even better, meaning you can give different subjects different stimuli, right? So if you have like a pool of 1,000 stimuli, 50 subjects, great, give each subject 20 different stimuli instead of giving everyone the same 20, and then your test statistics will be pretty much fine. So that's the moral of the story, and I'll stop there and take questions. Yeah, Satra.
So in that model, if you did still treat it as a fixed effects model, but had each stimulus or each group of stimulus of the same stimulus be a condition, how would that change?
Doesn't work. So it's better in the sense you're capturing variation, but the big problem is you're not capturing the random variance component, right? So you're not modeling the uncertainty associated with sampling randomly. You're basically saying now there are these exact 12 things we care about, but you're not treating it as if there's some underlying latent thing you're sampling for. So it's a little bit better, but it doesn't go near. And we have a model that looks at that as well, and the answer is it's intermediate, but much closer to the sort of standard model, and it doesn't solve the problem at all. It doesn't adjust the uncertainty here appropriately. Yeah, that would be nice because then it's easier to fix. In the first level models, you just add all the stimuli, but it doesn't work. And that's what makes it hard, is that you need to fit a model that has both of these things simultaneously, right? So you need one big model in some sense. Although there might be potentially ways to approximate that.
From an experimental design perspective, does this sort of speak to the thought that we should put when we're choosing our stimuli what distribution we're really sampling from? Right? Like if we want emotional faces, should we be thinking about the prior phase rate for the frequency with which people are going to encounter negative faces of a given token?
Yes and no. So well, definitely yes, but there's two separate issues, right? The issue of do you want to make an inference over any population, which is separate from the question of what is the population you're drawing the inference of. So this does not, doing this does not mean that you get to generalize over whatever's in your head. So you might want to, you care about emotional versus non-emotional faces. Modeling stimulus as a random effect here doesn't mean that you can draw that conclusion. All it means is that you can at least say for the particular population that your stimuli were sampled from, right? You can generalize over those. Yeah, and so you definitely want to, right? You want to think about the sampling strategy, but you also want to think obviously about the construct validity. I mean, is that population even what you want? The latter problem is kind of insuperable in the sense that there's generally, there's psychometrics exist in some sense to solve that problem. But at bare minimum, we should be modeling the stimuli we are sampling as if they were sampled, which they are, right? I mean, there are some, but it's very rare to actually care only about the stimuli you used in your experiment. Just like you don't model subjects as a fixed effect because you care about people like the ones who came into your experiment, not just the 3 people that you scanned. Luke?
So this isn't super well thought out, but let's say, the example you have is faces, and you have the same face that's making a fear expression, anger expression. You treat those as 2 independent distributions, or you
So, well, it depends on the design. I mean, so you're going to be much better off in general because then you only have problems if there's an interaction, right, between the stimulus and the condition. And that will happen sometimes. Most of the time, that variance is much smaller. So another lesson to take away from this is a good point is if you can, cross subjects with stimuli so that every subject gets, or sorry, with conditions, so that every subject gets every stimulus is in all of the conditions. But it's not going to solve the problem. I mean, you will have situations where there are stimuli that produce a strong response in one condition but not another. But it's definitely helpful if you can't model it, then do whatever you can to. So lots of stimuli and try and cross condition. Just like and within subject designs, we try to cross condition and subject, same principle. Yeah.
A very naive question. Does this apply to identical, very basic sensory stimuli?
That's a good question. I don't know. I mean, I think it would depend on what you think of as the, so, I've thought about this in the context of pain, for example, right? With like painful stimulation, it's not clear you want to treat like, a 45-degree thermal stimulation as like drawn from a population, 45, 46, 47, they're not drawn from some population of, infinite values. So I think it depends very much on how you think about it. One way to think about it is like, do you want to make predictions for new values that aren't just, interpolated in between these values, but are in some sense could be qualitatively different, but drawn from the same population? If the answer is yes, then you want to model as a random effect. If the answer is no, then you're fine not modeling that. But it depends on the domain. Most of the time, I would say, though, you probably do want to do the bottom thing. Okay, I should probably stop there. Yeah, we can.
Well, let's move on. Thanks, Tal.
That's all right, man. I'll take it.
All right, 5 minutes, 20 slides. Okay, I brought this up earlier, so I think I use this as a propaganda machine to try and get more and more people to do annotation. So hypothesis is a way of annotating the web. And how do you do it? Well, you go to some place like OpenFMRI, and luckily someone has already done this, so I don't actually have to do it. Oh, it's Tal who did it yesterday. So yesterday this thing came up, how do we recognize data sets that are out there? So Tal went in and used the structured form that was posted on our Slack channel to annotate it. Okay, so essentially you go to Hypothesis, you sign up, you download a Google Chrome extension, and then from that point on you can do annotations. What can you do with it? You can actually look at tags like NHW16, which gives you all the people who've tagged things using NHW16. You can also write code to export those tags in a bunch of different formats. So it's annotations that you can extract out, which I don't think is completely possible with Twitter, but not fully. So it's a way of also creating public things. The other thing that you can do, you can annotate PDFs. So this is a PDF on archive, and I can see that somebody has gone in and annotated it. So I load it up, and lo and behold, as soon as it loads, there should be a ridiculous number of annotations. Might have been Marianne who did this, who's also trying to promote this in various places. But the thing is, you can annotate PDFs, you can annotate web pages, and you can share those annotations. You can create private groups of annotations. You can create self-annotations, all within a browser-based framework. Okay, that's part one. Part two, for those who don't know, many people in this room I know of Neurostars. It's a framework like Stack Overflow for asking questions and answers related to neuroscience. So for those who were thinking of where to ask questions that you come across, this would be a good place. It's used a lot for a variety of neuroscience software, but it's a general place for asking questions that you may want the community to answer. All right, part three, and
Can I say one thing?
Yeah.
One really cool way to use this is like you have some software and you don't want to deal with like a mailing list or something like that. Like we do OpenFMRI support up here now, and it's a great way to like have kind of a community support software.
Great, yeah. Third piece, many of us build or do things in the app space. So Ionic is a framework that allows you to write JavaScript code that compiles down to native iOS and Android apps. So we use that framework. Actually, Apple created this framework called ResearchKit for generating research app, but it's iOS specific, so you have to be stuck to Apple's platform to use it. Luckily, Nino, who took Ionic and built Ionic ResearchKit, which allows you to do ResearchKit-like apps in Ionic. Well, we took that and we built a framework on top of Nino's framework, which allows you to generate apps, but you do this through a specification that's a JSON file. So in a JSON file, you specify, so this is a consent section. The spec comes down from a certain URL, and you can have different things in a consent section. You can even have consent review, so you ask people questions after the consent section to make sure they actually read the consent before signing, if you want to. Then there are things like eligibility, if somebody can sign up for this study or not. And then finally, you have surveys. And each survey has a cron job likes date stamp, which says when these questions could be asked. So you could have a longitudinal app, and you can specify, I want to ask this question every Tuesday or every first of the month. And each survey question is expressed as an Ionic ResearchKit thing. So in this case, this particular thing is a voice recording, and we record with a particular duration. And then the final thing is, this data all goes up into a Mongo database called Girder, developed by Kitware. And so you can see that the responses, so I can click on this, and this at least contains all my responses and the JSONs uploaded. So all of this is openly available that people could use to develop their own apps and do things. I'll stop there.
And after this last talk, I'm just reminding you, there will be a breakout session here with Tara about Chris, Mike.
Okay, cool. So I'm going to talk about very briefly about a new iteration over OpenFMRI. I've been working for over a year now. Who heard about OpenFMRI? Okay, that's good. It's a repository of public MRI data. And we were thinking a while ago, how can we make it better? How can we incentivize people to share data, and what can we give back? And we thought that the best way of doing that is to provide data analysis as a service, as a free service. So we also took this opportunity to rebuild the whole thing and make things a bit easier. So this is it, and you can later come to me or email me. I can give you access to a private beta so you can play around and try to break it. So you log in with your Google account or something similar. And you immediately have access to your data set, or you can go to your to the public data sets, or you can just go and upload a data set. And right now you get in touch with the data curator, they send you a Dropbox link, and it's basically an involved process. But now we facilitate on top of the bits standard, which I'm going to talk about more tomorrow. So things are a little bit easier. So I can just go and select the data set from my hard drive. I'm going to use something small, so we wouldn't look at progress bars for long. You give it a name, Neurohackweek for the win. And then the validation process happens. And as you can see, the validation, the progress thing was very, very fast. And why is it so? Because it's not done server side, it's done on the client side. So everything is written in JavaScript, and the validator for bits is also a standalone app. You can use it for your data set without uploading it anywhere if you want to use it in your lab. And it gives you errors, but also warnings here. There's no slice timing for this data set. And I actually don't know what the slice timing is. It's an old data set. We don't have this information. But it's not a deal breaker, but some things will not work in the future. So I continue.
And you can see that the data set is being uploaded. And I can go and do this. Because I ran out of battery and I don't know, I had to go. So, well, there you go. There you go, that's my source. So we support basically interrupted transfers. So I can go back and here it will tell me, oh, you interrupted this transfer, and I can click resume. Because those data sets are usually bigger and things happen, so you want to make that easy. All right, cool. So I do continue, and stuff resumes. Yep. So what can we do with this, with these data sets? Let's have a look at this one. I already created it, and I can. What I can do is. I can publish it, they're originally private, but I can also create something called snapshots. So I can, let's say, modify it and add another character, Joe. I can also add files, modify files, remove files, add subjects, remove subjects, and then because we want to make everything reproducible, I have to create a snapshot of this data set before I can run an analysis. So when I run an analysis, I can always go back to the origin of the data. So when I have a snapshot here, I can click Run Analysis, and here I can select a task. And those tasks, we encourage all of you and your friends, family, colleagues, strangers on the street to come to us with your methods, with your tools, with your pipelines. We're especially interested in robust pipelines that go from zero to hero, but also at the higher level analysis tools as well. And we can provide them as a service for you to a bigger range of users than you would get if you would just basically account for people downloading, installing, and etc., etc. So let's have a look at such app. So here we have FreeSurfer, for example, and each app has information about the acknowledgement, so who you should cite when you use this. So we are all about giving back. So you're gonna get the credit that's important. Also, like how to get help, choose options, etc., etc. So I can go and start this this up, and demonitis happens. So I'll just start and try something else with the example app. Who knows? Yes.
Choose subject.
By default, all subjects should run. So I'll just okay. So we're testing different backends. So because we don't want to, run out of steam and resources straight away, we are using high-performance computers available at actually 2 universities right now to run all of this. We can switch to cloud with not much effort, but it's right now a bit more expensive. I'll talk tomorrow how to get your method and turn into a bids app, which involves the skills that we've learned in the very first day. But this is basically it. This is our vision of next version of OpenFMRI. So get in touch if you want a demo. Get in touch if you want to deploy your tools here. And thank you very much.
And with that, we conclude this session of lightning talks. Thanks to all the speakers again.